So it'd be a little bit of a change of pace right now is that, there we go. I'm a molecular guy. You guys have been talking about epidemiology and treatment and stuff like that. Well, I'm a molecule guy. And so I'm going to tell you a little bit about the coming molecules and what we need to prepare for to be able to bring them to Africa where they're so desperately needed. Okay, how does... Okay. How many professors does it take to advance slides? It's a trick question. Okay, that's what I've been pushing. There we go. Thank you. Okay, so don't need to review this. You guys heard this quite a great deal. The things that I want to point out here is that therapy does not cure the infection. Okay, we can slow down the death rates, but we can't cure the infection yet that's driving the disease. And curing HBV is going to require new drugs with new molecular targets. Okay, so hepatitis delta virus, we haven't spoken about quite as much. Molecularly, it's a fascinating molecule, but I really would not want to have it in my liver. It's a subviral RNA pathogen. Its closest relatives is actually things like plant viroids. There's lots of them in potatoes, for example, petunias. That's what it's molecularly most closely related to. But it's a parasite of HBV and requires the surface antigen of HBV to get between cells. It infects around 1.2 million people in Africa, and it's usually acquired as a superinfection after being infected with HBV as birth. Having hepatitis D is the worst form of viral hepatitis out there. Many of you in this audience know this very well. There's no highly effective drugs for HDV. Pegylated interferon is used, but it's really not very effective. And the nucleoside analogs that can control B don't really affect D at all. And so again, we're going to need new drugs with new targets, and that's the area that I am more familiar with. Okay, so I'm going to spend just a couple of moments talking about the new classes of the therapies that are under development. Each one of those bullet points I could give an entire seminar on. So I'm just going to go over them really briefly. If anybody has questions about it, just hunt me down over coffee, and I'm happy to talk about it. So for HBV, most of the ones that are coming up at this point are additional classes of viral replication inhibitors, such as the capsid assembly modifiers. These things have a great deal of promise in improving control of HBV compared to the treatment with the nucleoside analogs. There's also many more interesting versions like RNA interference and oligo-DNA nucleotides, entry inhibitors, therapeutic vaccinations. The one that I have just gotten really excited about recently at the HBV meeting in Berlin last week was genetic editors. For a long time, I was very, very down on those babies, but it's looking like we may actually get to the point where we can get a 99% deactivation of the viral cccDNA, which is the molecule in the nucleus we need to destroy, with a single injection in some cases. They're getting that in animals now. And they've figured out the delivery problems, which was what was really bothering me for a long time. So I'm really excited about the genetic editors right now. There's also monoclonal antibodies coming along that are very promising. Cytokines aren't really making much waves in the water yet, but we're hopeful. And then there are secretion inhibitors for the surface antigen, which are there. HDV, we've got bigger problems. There's very little investment going on there. We've got entry inhibitors. Bulevirtide was approved. That's an injectable drug, and so that's got delivery and deployment problems. But there's many of them, small molecule, orally available drugs that are looking really good in the late preclinical and early clinical stages right now. So I do think those are going to come along well. Prenylation inhibitors are a problem. We've had one drug move forward, but I'm nervous about that one. Of course, monoclonal antibodies against the HBV surface antigen work very well against hepatitis delta virus also because it is coded by the surface antigen. And it's also important to note that curing HBV will eventually cure HDV because HDV is dependent on HBV. Okay, so let's get to the primary point of this talk is what's the problems with trying to advance cures in Africa? We've been talking about treatments, with using current treatments, but let's talk about the challenges to curing, and they're all the ones that we've just been talking about, but they're worse because, of course, you've got more expensive, more complicated therapies coming down the lines. The ones that I want to focus here is on the enormous diversity within the African continent. The linguistic, cultural, and geographic diversity is absolutely mind-boggling. And the other one that's really important here is human genetic variability. 90% of human genetic variability is found in sub-Saharan Africans. All of us, from Europe or America or something like that, we form a really tiny slice of the genetic variation found in human beings. Most of those don't matter other than the fact that it makes me not look like our distinguished chairs here. Okay, so what? But what does matter is some of those variations affect immune modulating or immune controlling genes. Some of them affect the transporters that transport drugs into or out of a cell. The CYP450s that are involved in destroying the drugs. So those are subject to the variations in the human genetic population also. And that affects drug metabolism, which affects dosing. And so it's extremely important that those sorts of issues be understood before we start rolling out drugs that were developed by Gilead in the United States to sub-Saharan Africa, because the dosing regimens may not be the same. Okay? Another thing is high HBV and HDV genetic diversity. These are very genetically variable viruses, and just out of chance, the field has developed that a certain very limited number of strains are worked on in the lab, including my lab. I'm guilty of it also. But they don't match the strains and genotypes that are found in Africa. So we don't know if the direct-acting antivirals are necessarily going to work against those strains as well as they do against the strains for which they're being developed. And, of course, the other issues that we've been discussing quite extensively. Oh, come on. Okay. Could you please advance the slide? There we go. Okay. So what can we do right now? Well, here it's time for some more depression. Okay. Unfortunately, we've got time to work on this problem because the widely curative therapies are not imminent. Okay. They are coming, but we're talking on the order of a decade, not next week. So the examples, we're making great progress. The current best curative therapies are running around 40 to, in some cases, 50% cures in carefully selected populations in small trials. And so that's a lot better than the low single digits that we used to be doing, so I'm happy about that, but it's not anywhere near the 95% plus where we need to be. So what can we do right now? We've got to hold the line, as we've been talking about. We've got to hold the line. We've got to control the disease until these newer therapies are online and are made affordable enough to be used here. And so increasing the birth dose HBV vaccination rates is critical. People have been talking about that all morning. That's good. I am absolutely ashamed to say that my country is in the process of almost certainly removing the birth dose recommendation due to the Luddites that are running the U.S. Public health system right now. I am absolutely ashamed to admit that to this audience. The other thing that we can do is really expand the use of tenofovir and entecavir whenever possible to slow disease progression. And I know there's all sorts of problems in executing that, but it will really help. One of the things to point out is when the recommendations are developed, there's a large body of data that is not looked at because it is not considered of the type of data that is typically used for guiding medical decisions. But they are very solid data, and they discuss the mechanisms of pathogenesis. And those data will never appear in a classic high-quality clinical study designed to test their effects, because it takes 30 years to run the clinical study, and it is logistically and ethically unfeasible to conduct those studies. But the basic science data screams loud and clear: treat people earlier, treat more people. So ICE-HBV, the International Coalition to Eliminate HBV, just reviewed the evidence for this, and we published 2 articles in Lancet Gastroenterology just a little bit ago. They aren't even out in the paper version of the journal yet. But if you want to see a copy of them, either click on those links or give me a call and I'll email them to you. Okay, so medical research opportunities to accelerate cure in Africa. Much of these we've been talking about, strategies to improve both birth vaccination rates, conduct PK, pharmacokinetic studies, and phase 1 safety trials in African populations to ensure that these experimental drugs work in the African populations as they do elsewhere. We need to map the HDV and HBV genetic diversity more thoroughly than we have done. And for those molecules where we know where they're binding on the various viral proteins, flag variants that could alter sensitivity for closer testing. And then we need to, another thing that I'm really excited about would be rigorously test traditional African medications for efficacy. People have been using all sorts of things for medicines for a very long time, and sometimes they work. Tamoxifen, a drug used for breast cancer, was developed from a traditional cure. And so testing those may help launch very productive drug development campaigns. We need to develop our scientific and medical workforce, and we need to ensure that the diagnostics and therapeutics can be delivered effectively. Okay, and then public health opportunities, again, improve diagnosis rates. We can't treat people if we don't know they're infected. We need to improve the education. Again, we've been talking about this all morning. Expand vaccination, fighting stigma, and I have absolutely no idea how to do that. But those of you who work in those areas, please work hard at it, because it's really, really important. Ask infected people what they want out of therapies, and share that information with the drug companies. Develop advocacy and community support organizations. Kick up a stink. We needed that political support that people were talking about. Well, that's the way it comes into play. Lobby for international support. Unfortunately, things are going the wrong direction. And coordinate with HIV control programs, and I was delighted to see that there's very aggressive action on that going on. So to conclude, is there an opportunity for hepatitis B and D cure in Africa? And yes, there is. I strongly believe there is, but it's not going to be fast, unfortunately. So what we need to do is vastly expand the diagnosis rates, prepare for the upcoming curative therapies, and hold the line as best as we can to reduce the rates of disease by expanding vaccination and improving treatment coverage rates until those new drugs are ready and we've managed to suppress the price down to the point where they can be delivered. So thank you.